No Entrevistando Cientistas de hoje, nós vamos entrevistar a doutora Renata Pasqualini. A doutora Renata Pasqualini é pesquisadora internacionalmente reconhecida por seu trabalho em target molecular no câncer. Ela é graduada na Universidade de São Paulo em Ciências Biológicas, onde também fez o seu doutorado em bioquímica com parte da pesquisa desenvolvida no Institute for Cancer Research e pós-doutorado em instituições como Harvard e Dana-Farber Cancer Institute. Atualmente é professora do Rutgers University em Nova Jersey, nos Estados Unidos, e lidera pesquisas inovadoras que unem biotecnologia, terapia gênica e modulação do microambiente tumoral.
Gostaria de começar nossa entrevista relembrando seu artigo de 1996, no qual você introduziu a técnica de in vivo phage display como uma estratégia inovadora de targeting vascular. Como surgiu a ideia de aplicar essa técnica diretamente em organismos vivos? E como ela deu origem às abordagens que seu grupo desenvolve hoje, voltadas ao direcionamento preciso de células do microambiente tumoral, como o endotélio glioblastoma e os macrófagos associados ao tumor no câncer de mama triplo negativo, para induzir a apoptose seletiva como estratégia terapêutica?
So in vivo phage display was again invented, perfected, and developed through time, again, since the mid-90s. So it's important, I think, for us to talk about phage a little bit, which is the system that we use for both discovery and therapy in certain instances, and is the basis of the papers you happen to select to discuss today. So phage is a virus. It's the largest biomass in the planet. There's tons of phage in the oceans, everywhere, multiple types. And it's a fascinating virus because it evolved through time to infect only bacteria. So it has no tropism whatsoever towards mammalian cells or any organ in the body, unless you engineer the capsid to display a ligand that can be either an antibody or a peptide. So a scientist by the name of George Smith, who happened to have won the Nobel Prize a couple of years ago, engineered this virus for you to be able to display a library of compounds of different binding capabilities in the capsid. So you could have as many as 10 to the 9 permutations of a peptide combinatorially displayed. And what you could do with these libraries is to map protein-protein interacting sites. So if you had a receptor immobilized in a plate, you could do a biopanning and identify specific phage that would bind to that protein. So the inspiration for the in vivo phage display technology was at the time, there was a lot of attention being paid to blood vessels, angiogenesis. Judah Folkman was just coming up with this idea that tumors needed blood vessels to grow and the like. What we were doing was just trying to understand, again, the process of site-specific metastasis, and also understand the fact that it was expected and acceptable that tissues would carry unique marker molecules that would be accessible to circulating ligands. The reason for that is cells and proteins that are circulating need to know where they are. Am I in the lung? Am I in the kidney? Am I in the brain? And need to know where to go in situations of inflammation, cancer, trauma. So we figured there must be a zip code system in the vasculature, but nobody had a way to find those receptors because you need to find them in vivo in a microanatomical context that's relevant. That's when we had the idea to inject the library of phage systemically, allow the phage to distribute to all the tissues. And then after the library cleared from the circulation, we could recover each tissue and sequence the phage that homed there. And through subtraction and bioinformatics, we could say, well, this set of phage go to the brain, this go to the lung, this go to the kidney. And from the ligands, we could then use standard biochemistry to purify the receptor, namely the zip code that was mediating the homing. So then you had the ligand receptor pair that would allow you to localize any payload of interest that you could conjugate to the ligand or the phage itself to deliver something of interest, a therapy, an imaging agent, a nanoparticle. And this was the genesis of the discovery. And then when we left California, the Burnham, we were recruited to the MD Anderson Cancer Center in Texas. It's a huge cancer center, the #1 in America. We were able to translate in vivo phage display to patients so that we could do these screenings directly in patients. And this was very important because it accelerates drug development substantially. If you want to target a therapy, you want to know that it will work in patients and how best to find out if you do the screening directly in patients. So there's these two aspects for this technology that we find very rewarding. First is the discovery of these markers that are often very new. Nobody knew they would be important in multiple diseases or normal organs. And also the translational implications for the targetability of the ligands in terms of specific therapies, gene therapy, chemotherapy and multiple modalities that we have been developing over the years.
No uso dos vetores AVP, o gene suicida HSVTK permite a indução de apoptose seletiva no microambiente tumoral, especialmente após a administração de ganciclovir. Como essa abordagem funciona desde o reconhecimento do alvo até a morte celular? E o que torna essa plataforma uma opção terapêutica promissora frente a outras tecnologias?
So as I was saying, first you do the targeting, you find the ligand, you find the receptor, and you know it homes to the tissue or organ you want. And it's important to emphasize that there's several levels of diversity in the vasculature. One is the normal organs that have their zip codes. And then there's a whole new level of diversity that comes up or emerges when you have a disease process going on, including but not limited to cancer and angiogenesis, which is the abnormal proliferation of blood vessels that brings new markers that can be specifically targeted. So if you use tumor targeting phage to deliver something to a tumor, you can leverage this in two ways. You can use the peptide or the antibody linked to a drug or an imaging agent. But another interesting aspect of phage is that the genome is very easy to engineer. It's a single-stranded DNA genome. It's not very big. It's about 10 kb. And you can introduce elements in that genome. So the capsid stays the same, targeted in the way you discovered it in the first place, which is the ability to home to tumors specifically and nowhere else in the body. But then in the genome level, to make these AAV phage vectors, what we did was to introduce AAV elements. AAV is another vector that's single-stranded, but the capsid is very problematic because it always ends up in the liver and transduces other tissues that you would not want to do. So specificity for targeting in gene therapy is a big unmet need. So this is why we decided to bring together the best of both worlds. What is one of the best gene therapy vectors? AAV, but it's not targeted. Phage is targeted, but if you clone a mammalian promoter and a gene, it doesn't make for a very good gene therapy vector. But if you introduce the ITRs from AAV, the inverted terminal repeats, then you create a very potent gene delivery tool. Because what happens is the phage homes to the tumor vasculature and the tumor cells that express the zip code, and then it gets internalized specifically, and then the genome is released inside the cell. And the AAV genome forms concatenators, which fire very potent gene expression. And this way, you get the gene expressed only in tumors and nowhere else. And you mentioned TK, thymidine kinase. This is a fascinating gene because it has dual function. It can be used for imaging and it can be used for therapy. So if you express thymidine kinase only in tumor cells and tumor vasculature, tumor infiltrating cells, in the case of macrophage targeting, as we will discuss, you will have TK only on that site and nowhere else. And you can image if you give a tracer like FIAL that will light up cells that are expressing TK. And if you give ganciclovir, which is another substrate of TK, you induce cell death very potently and also have a bystander effect because, again, the substrates diffuse and kill adjacent cells through gap junctions and also induce an inflammatory response that also induces killing. So you have a bystander effect. It's obviously important because it's targeted.
No câncer de mama triplo negativo, os macrófagos associados ao tumor do tipo M2 promovem um microambiente imunossupressor. A depressão desses TAMs por meio do targeting de PDI-A3 gerou uma mudança inflamatória no microambiente tumoral. Essa estratégia poderia ser suficiente para conter o crescimento tumoral ou seria mais eficaz em combinação com outras terapias?
So, in the work you're talking about, it was a very interesting departure from a lot of papers that we had published before, which had to do with phage. In this particular, and remember, the screenings are done blindly. So when you recover the phage, you really don't know where they're homing to in the beginning. So once you study and you find out where is that particular phage homing to and which cell is it targeting and internalizing into, then you can understand the mechanism behind the targeting. So in this particular study, which was very elegant, for the first time, we targeted tumor-infiltrating macrophages, which happen to be macrophages that have pro-tumorigenic activity. This is why if we induce cell killing within this population of cells specifically, you observe tumor regression, which is a very nice finding view. Now you bring up the aspect of therapy. Is this really a therapeutic modality that would be extremely efficient as to be compared to, for instance, antibody-drug conjugates or CAR-T? And I don't think that's well characterized yet. But if I were to guess based on the literature, I would say this would be more of a combination therapy approach, that you would use the destruction of these tumor infiltrating cells to facilitate the activity of other more potent cytotoxics.
Considerando os avanços atuais em medicina de precisão, imunoterapia, entrega molecular, como você enxerga o futuro das terapias antitumorais baseadas em target seletivo e indução de apoptose? E quais são, na sua visão, os principais desafios para levar essas estratégias à prática clínica e como as abordagens que o seu grupo vem desenvolvendo se inserem nesse cenário em evolução?
So when you start targeting a tumor, as I mentioned before, with a library that's random, you can obtain phage particles and ligands that are displayed by this phage that recognize receptors that are in multiple compartments. So we have different scenarios that we observed over the years. Certain ligands are very interesting because the receptor is present both in the vasculature, in perivascular cells, such as pericytes, in the tumor cells, and in infiltrating cells. So those make for ideal targets because you hit many, many birds with one stone. And what we've been developing and prioritizing in the laboratory in terms of drug development programs that are already, some are already in the clinic, they explore these types of receptors that are highly upregulated or exclusive present in tumors, expressed in multiple compartments, and they can be used for the conjugation of cytotoxics in the case of ADCs, antibody-drug conjugates, or in the case of peptidomimetics that carry a propoptotic entity. So as part of our work, if you look back, we did carry out phase 1 trials in cancer patients that demonstrated the conceptual feasibility and to some extent the safety of the targeted therapies in these two formats, peptidomimetics and ADCs. Now, how they're going to turn out? Of course, it requires a lot of investment, it requires a lot of time, and it requires a lot of smart people to develop. This is why we were fortunate enough to have investors approach us to start 2 biotech companies that are now pursuing therapeutic approaches coming from the lab. One is called Embrace Therapeutics, and the other is called Phage Nova. So because what everybody's looking for are clean targets. Can I find a target that's only in the tumor and nowhere else? And I think this is where in vivo phage display may have a very important impact, so that you increase not only the efficacy, but also the safety.
Doutora Pasqualini, eu gostaria de agradecer em nome de todo o grupo por você ter aceitado o nosso convite. Foi um prazer poder entrevistar você hoje.
It was a pleasure to meet you and talk to you about our work. Thank you so much for the opportunity. Good luck, be good, study hard, and the sky is the limit. Take care.